Gram staining of bacteria. What is gram staining? Gram staining is a microbiological technique used to distinguish between different types of bacteria, based on their cell wall composition. It is a fundamental microbiology technique, used to differentiate bacteria into gram-positive and gram-negative groups based on their cell wall structure, aiding in the diagnosis and treatment of bacterial infections. Who discovered gram staining? Hans Christian Gram, a Danish physician and bacteriologist, developed the staining technique in 1884, which is still used today to differentiate bacteria into two major groups: gram-positive and gram-negative. What is the principle of gram staining? The gram stain distinguishes between two major groups of bacteria based on the composition of their cell walls. Gram staining relies on the ability of bacterial cell walls to retain a dye during the decolorization process. It helps to classify bacteria as either gram-positive or gram-negative. Gram-positive bacteria stain purple or blue due to a thick peptidoglycan layer in their cell walls, while gram-negative bacteria stain pink or red due to a thinner peptidoglycan layer and an outer membrane. Steps in gram staining. The gram staining procedure involves 4 key steps. Applying a primary stain, crystal violet. Adding a mordant, grams iodine. Decolorizing with alcohol or acetone, and applying a counter stain, safranin. Step 1. Primary stain, crystal violet. A heat-fixed smear of the bacterial culture is prepared on a slide. Bacteria are stained with a primary stain, usually crystal violet. The slide is flooded with crystal violet for about 1 minute, allowing the dye to bind to the cell walls. The slide is rinsed with water to remove excess stain. All bacteria are stained with crystal violet, a cationic dye that binds to the negatively charged cell wall components. This dye initially stains both gram-positive and gram-negative bacteria as purple. Step 2. Mordant, grams iodine. A mordant is a substance that enhances the binding of a dye to a material, in this case, the bacterial cell wall. After the primary stain, crystal violet, is applied, grams iodine is added. The iodine forms a complex with the crystal violet, which then becomes trapped within the peptidoglycan layer of the cell wall, especially in gram-positive bacteria. Grams iodine is applied to the slide for about 1 minute, creating a complex with the crystal violet. The slide is rinsed with water to remove excess iodine. This complex is then resistant to decolorization with alcohol. Step 3. Decolorization, alcohol or acetone. A decolorizing agent, such as ethanol or a mixture of ethanol and acetone, is applied to the slide. This step is crucial for differentiating between gram-positive and gram-negative bacteria. Gram-positive bacteria retain the crystal violet iodine complex, while gram-negative bacteria lose the stain, becoming colorless. The slide is rinsed with water immediately after decolorization. Step 4. Counterstain, safranin. Safranin, a red dye, is applied to the slide for about 30 seconds. Gram-negative bacteria, which are now colorless, will take up the safranin, appearing pink or red. Gram-positive bacteria, which still retain the crystal violet iodine complex, will remain purple. The slide is rinsed with water and dried. Results. Now we can differentiate bacteria into 2 major groups. Gram-positive bacteria. The thick peptidoglycan layer in gram-positive bacteria traps the crystal violet iodine complex, causing them to remain purple. Gram-negative bacteria. The thinner peptidoglycan and the outer membrane in gram-negative bacteria allow the crystal violet iodine complex to wash out during decolorization, leaving them colorless. The decolorized gram-negative bacteria are stained pink or red by the counter stain. Practical applications of gram staining. Diagnosis of bacterial infections. Gram staining helps determine if a bacterial infection is present, and whether the bacteria are gram-positive or gram-negative, which is crucial for selecting appropriate antibiotics. Healthcare providers order gram stains to help diagnose and treat certain bacterial infections, such as urinary tract infections and bacterial pneumonia. Guiding antibiotic selection. The gram stain result provides information that helps clinicians choose the most effective antibiotic based on the type of bacteria. Understanding bacterial morphology. Gram staining can reveal the shape of bacteria, like whether they are cocci or bacilli. and the arrangement of bacteria, which helps in identifying specific bacterial species. Research. Gram staining is a fundamental tool in bacterial research, used to classify bacteria and study their cell wall structure. Conclusion. And that wraps up our lecture on gram staining, a simple yet powerful tool in microbiology. It not only helps classify bacteria but also plays a crucial role in diagnosing infections and guiding treatment. Thank you all for listening, and I hope you found this informative and helpful. Stay curious, stay scientific, and see you next time.